listeners. Assalamualaikum warahmatullahi wabarakatuh, and a very good morning to all. Hope everyone will have a great time today, and especially for our last session of the International Summer School for today, which the first session will be guided, or the speaker itself is Dr. Pui Khoon. And then our second session will be about the virtual tour of Salak Ledok Nongko by Saudara Viko. So without further ado, before I invite our first speaker for today, I would like to share a little bit of her CV. So, okay, for Dr. Pui Khoon, she is a lecturer from the Industrial Biotechnology FIST, UMP. So her primary research basically designated on the adding value to underutilized food materials, food safety, and food product development. So she is basically an active member of the Drugs Discovery and Diagnostic 3D cluster. So Dr. Pui Khoon herself is a food science graduate from and then also from her master degree, and then she graduated for her doctoral degree at the University of Alberta, Canada. So if any one of you wants to know more about Canada, maybe can ask Dr. Hong. So for her expertise itself, mostly majoring in functional food safety, halal products, as well as antimicrobial optimization byproducts as well. All right. So that is the short CV of Dr. Pui Khoon. So without further ado, I would like to invite our speaker to give her lecture for the first session.
All right, thank you very much, MC Ms. Shafina. A very good morning to everyone. Welcome to the UNISA Summer School. This is the last day, so I hope we can take things a little bit take it slow a little bit, because this is the last day, so try to enjoy while we still have time. So I am actually sharing my screen right now. If my screen is visible to you all, can you please hit number 1 in the chat box again so that I can see that it's working? Okay. Yeah, because I'm using 2 screens at one time. So when I'm giving the talk, I think that I will disable the the camera. Just for the sake of convenience, I would say, because I'll be facing this direction. Okay, so where's my video? All right. Okay, so my talk today will be focused on omega-3 and omega-6 fatty acids. So I don't know if anyone ever know about this, but I'm sure that you will gain something towards the end of the talk, all right? So we have a few learning outcomes for today. I have 3 actually. So towards the end of this talk, I hope you will be able to describe the types of fatty acids which are functional ingredients, because most of the time you can see products in the markets, they actually highlight the content of omega-3 and omega-6 in the products. And the second one is, I hope you can differentiate between omega-3 and omega-6 fatty acids, okay? And last but not least, to understand sources, beneficial effects of polyunsaturated fatty acids, because omega-3 and omega-6 are regarded as polyunsaturated fatty acids. So before we kickstart, I also like to recommend students who would like to join a very short quiz session towards the end of this talk to download Kahoot. If you don't have the app by now, you can download it on your mobile phone. You just need to recognize this. This is the real Kahoot with 15 million downloads, okay? So this is the only one that we'll be using it for today, all right? So If you are ready, so let's go. So let's look at the chemical structures of lipids. Lipids in general, actually, it includes like the fats and oils, etc. So lipids actually, they exist in several forms. Let's look at the first form, which is the triglyceride. For triglyceride, how it is formed is actually by dehydration synthesis of 3 fatty acid chains. It can be different lengths, okay? So 3 of the fatty acids will join together to the backbone of glycerol, forming an ester bond in between. And the result is actually a triglyceride, and together with 3 molecules of water synthesized. Okay, so I want to give a question. This warming up. If anyone knows what's the opposite of dehydration reaction? Anyone? If you know the answer, you can type in the chat box. That seems like very quiet. So it starts with the word H. This is a hint. The first letter is H. Oh.
And okay. Yeah.
You're getting close. I'm not looking at hydration, but I can see there's one girl got it right. Just that a little bit of typo. Tibia, you got it right. It's actually hydrolysis. Okay. Oops. Sorry for my writing, but you get it. So the opposite of dehydration synthesis, actually, like you try to break it down so that you can get 3 different fatty acids. It's by hydrolysis because you need to use water again to break down the triglyceride. All right. So triglycerides actually, they are found as like mainly as storage fat in plants, especially in the seeds, okay, and also animals. Actually is under the skin, the adipose tissue. So most of the time you can see like the fatty acids, they come in even numbers because of the biosynthesis from acetyl coenzyme, where you have addition of 2 carbon at one time. So that's why you can see even numbers such as 16, 18, 20, and so forth. All right. So here's another look at triglyceride. I just want to highlight this is actually the ester bond. So when we want to hydrolyze the fatty acids from this triglyceride structure, actually we are trying to break the ester bond, all right, to yield the free fatty acid. Aside from triglyceride, as I said, we have another form of lipid, which is phospholipid. So phospholipid, it is actually has a phosphate group, which is polar. And you have, again, the fatty acid change at the other, I would say, at the other link, okay? You have the second and also the third link. It's linked with the fatty acid change. So most of the time we will write, we will draw phospholipid like this, because like we just want to symbolize that this is a polar head, which is hydrophilic. Okay, and then the hydrocarbon chains, which are hydrophobic. All right. So phospholipids are mainly found in the membrane of cells. All right. And another form of lipids is actually cholesterol, but I'm not going to discuss it for the sake of today's talk. But just for your information, cholesterol is also a type of lipid. So let's look at the types of fatty acids. I will start with a small structure, which we call short chain fatty acids, okay? Abbreviated as SCFA. So most of the time where we see fatty acids that contain less than 6 carbons, so we'll call them as short chain fatty acids. So most of the time they can be synthesized, okay, by fermentation of dietary prebiotics. These are actually like oligosaccharides. So they are prebiotics because they are not digestible, okay, when we consume food. So most of the time, prebiotics, you can get them from, let's say, vegetables, fresh fruits, etc. So, and milk as well, because human milk, we have prebiotics as well, but I'm not going to talk too much on that. Upon fermentation of dietary prebiotics, actually, we can get short-chain fatty acids. So the prebiotics that I'm talking about, we have this galacto-oligosaccharides, fructo-oligosaccharides, etc. So for short-chain fatty acid, it has a very close relationship with the gut microbiota. You see, the human gut has a lot of microbes, and it's actually the population could be like more than 800 bacterial species, and among them, they are good and bad bacteria. So the gut microbiota, we know that like the good ones, especially they are called probiotics. We want actually to keep them, okay, because they are related to the regulations of the host defense, whether it's immune or non-immune nature, because it's actually, some scientists even say that actually our gut is our second brain, but well, we still have many things unanswered, because like what happens in the gut, yes, actually, like it's not just affecting our physiological functions, it's also affect our mood as well. So all this, it has something to do with the metabolites, including the fatty acids that we have in the gastrointestinal tract. So just for the sake of short-chain fatty acids, let me just go back to our topic. So like the types of short-chain fatty acids generated by the microbiota in our gut includes acetate, propionate, and butyric. They are very short. These are like 2 or 3 and also 4 carbons. All right. So how do gut microbiota influence, okay, our physiological functions? Actually, like when they are sitting in our GI tract, actually they will produce metabolites, which I'm talking about, like let's say short-chain fatty acids. For example, it can directly affect our physiological activities, or indirectly, actually, we can harvest it when we do some sort of like fermentation, and we can incorporate that into our functional foods, and we take the food, and we can actually affect changes in our physiological functions as well. All right. So the main short-chain fatty acids that I mentioned, they are acetate, propionate, and butyric. So these are their respective functions, but I'm not going through them one by one, but I think you can read. So mostly, like for acetate, is focused more on the muscles. Propionate is focused more on the liver metabolism, and butyric is in the chorionic epithelium. All right. So let us move on to MUFA, which is slightly longer than SCFA. So MUFA actually stands for, everybody, mono unsaturated. Where's my U? Okay, mono unsaturated fatty acids. MUFA. So MUFA, as the name suggests, there's one unsaturated bond, which is one double bond. So here I have one example for you. The common name for this particular structure is called oleic acid, which consists of 18 carbons and one double bond. Well, this is actually a, what do you call this? Shorthand nomenclature suggested by the IUPAC. Okay. So we use this system to identify if we are lazy to write the name of oleic acid, we don't want to write it, we can write it in numbers like this. Okay, this is called shorthand nomenclature, suggested by IUPAC again. So by reading this, we know that this oleic acid actually contains 18 carbons. You can count from here to here to here to here. You have 18. All right. Don't believe me. Let's count together. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Sorry. 1, 2, 3, 4, 5, 6, 7, 8, 9 here. And then you have 10. Okay. Where is it? 11, 12, 13, 14, 15, 16, 17, 18. All right. So when it's at the, what do you call this, 9 position, so there's one double bond. All right. So how do we write it? Is 18 colon, and then we will indicate the number of double bonds. In this case, there's only one double bond, so there's one, and this is actually like we can replace it with the omega sign, or we can use N as well. So N indicates where the first double bond is detected at. So in this case, it's at the 9 carbon. I don't know why my pencil is not working well today. So anyway, one more time. Okay, so it's, excuse me for a while. So there you go. This is carbon number 9. All right. So therefore, this whole thing summarizes there are at least 3 information. There's one double bond over here, and then the first double bond is detected in the 9 carbon. Okay. From the omega N. Omega N is actually here. So we count. This is number 9. So we always go with the methyl N. Okay, for omega system. And 18 actually is the length of the carbon. So there you have it. Why we call it omega 9? Because the first double bond is detected at carbon number 9. So since I'm talking about C18 fatty acid, might as well just mention the saturated one. So saturated fatty acid, we have another, like this is its brother, which is 18-0. So 18-0, this is stearic acid, also known as octadecanoic acid. So because it has no double bond, so therefore we put it as 18 colon 0. All right, because there's no double bond. It looks something like this. Okay, and then of course you have the H here. All right, so if you're good, then we can move on to the next slide, because if you are still confused, you can raise hand. I will explain one more time why, because we will have a quiz that will test your understanding by looking at diagrams like this. Okay, I think it's very straightforward, I guess. So let's move on to the next slide. Well, it's still a continuation of monounsaturated fatty acids. Just want to tell you, in nature, everything comes in a package. All right, so you have a mixture of things. For example, here I have a table contains the shorthand nomenclature of each MUFA. Okay, you have the palmitoleic acid, myristoleic acid, oleic acid, etc. And it is actually listed based on the composition of different oil. For example, we have palm oil. I guess everybody knows what palm oils are, right? Kelapa sawit. What do you call palm oil in Indonesia? Is it kelapa sawit as well?
Yes, kelapa sawit.
Okay, thank you very much, Ms. Shafina. I was actually expecting students to answer me to see whether they are awake or what. Anyway, so coming back to palm oil, as I said, everything comes in a package, in a mixture, in nature. So we have actually combination of different MUFA in palm oil alone. So palmitoleic acid is not just exclusively found in palm oil, it's also found in peanut oil, for example. So You can also have a look, you can ponder upon, I mean, like you have this stare at this table for a few seconds, like actually have in order to, if you analyze the content of MUFA in sunflower oil, you also notice that there are different types of MUFA present in sunflower oil. Okay, so all this, this is very easy to read, but just for well, it's not mentioned though, like what is the first double bond is detected. But anyway, just to tell you, like this is like 16 carbon, 17 carbon, 18, and so forth. Okay. If you are good, so let's move on to the next slide. So here, this is polyunsaturated fatty acids. So the word poly means it's more than one. Okay, so more than one unsaturated bond. So therefore, the name polyunsaturated fatty acids, PUFA. Normally, we say PUFA because it's easier to mention. So PUFAs actually, they have, like I said, more than one double bond and is separated by a single methylene group. We'll see the diagram in a while, don't worry. But most of the time, the PUFAs that we focus on, actually, they are three. They are the omega-3, omega-6, and omega-9 fatty acids. The essential ones, however, actually, they are the omega-3 and omega-6 fatty acids. Anyone knows why? Maybe you can type it in the chat box. The hint is we are lacking of certain enzymes. Lack certain enzymes. Pardon for my writing, but I think it's still readable. Lack certain enzymes. So what are they? I'm not so sure whether you learn it in biochemistry, but another hint. This is delta. Okay. I will give you the answer in a while. All right. We have like we are lacking two enzymes mainly in our body that will refrain us from making omega-3 and omega-6 in our body. So that's why they are regarded as essential fatty acids. So omega-3 and omega-6. We need to get them from our diet. All right. So the essential fatty acids and omega fatty acids, especially linolenic, the omega-3, and linoleic acid, they are very, very essential. I just want to stress here, okay? So I put two ticks here. And other essential fatty acids, and they include eicosapentaenoic acid. I know it's a mouthful, but you need to know their name by heart because it's just like memorizing somebody's name, right? So spelling everything, you need to make sure that it's correct. And docosahexaenoic acid from omega-3. For omega-6, you have arachidonic acid and docosapentaenoic acid. All right. Okay. So let's move on. Somebody's left in the waiting room. Should I admit or what? Okay. So let me just, okay. So here we have the important omega-3 and omega-6 fatty acids in this diagram. So let's go through their structures one by one, shall we? So the first one, how do I get rid of this thing on top? Okay, I just don't want to touch it then. Omega-3 fatty acids, you have this particular structure. So let me just bring the magnifier. I think it's better. So here you have omega-3 fatty acids. Okay, the first one is called alpha linolenic acid. So somebody. Okay. So for alpha linoleic acid, this is a C18, 18 carbon fatty acid with 3 double bonds. This is a omega-3. Okay, so how do we write it out in our diagram? So for omega system, we always begin from the methyl end. So CH3 is methyl, so this is the first carbon. So this is like 1, 2, 3. So the first double bond is detected in the third carbon. So therefore, this is omega-3. Okay. So the rest, they have the similar arrangement. Okay. And then you will have different types of different numbers of double bonds recorded. For eicosapentaenoic acid, penta is 5, eicosa is actually 20. All right, so it's 20 with 5 double bonds. So therefore, it's 20 colon 5. Okay, as the chain gets longer, you have 22, 6, which is docosahexaenoic acid. So this one has 1, 2, 3, 4, 5, 6 double bond, if you can follow me. Everything is good for omega-3. If it is okay, please put 3 in the chat box so that I know that you are following me. Only tibia? Okay, thank you. Get the 3 coming. Only a few respond. Okay. At least I know like some people are listening. Okay. So let's move on to number 2, which is omega-6 fatty acids. So for omega-6 fatty acids, we have linoleic acid. Oh, I forgot to mention about the short form. Let me just go back. So for alpha linoleic acid, we have ALA. Okay, we can write ALA, or we can write the full name such as alpha linoleic acid. And for EPA, this is for eicosapentaenoic acid. The same goes to DHA, which is for the docosahexaenoic acid. This is understood, all right? Because well, if you go to the market, you can see people always mention about EPA, DHA. They are talking about this one, okay? When it comes to fatty acid, okay? We need to ask them, are you talking about fatty acids? Yes, the EPA, the DHA. For omega-6 fatty acids, we have LA. So please do not get confused. For omega-3, you have ALA, all right? For omega-6, you have LA, that is linoleic acid. This one is linoleic acid, okay? Because a lot of students got it confused for the first time. So for omega-6 fatty acids, you have linoleic acid, which is C18, 2 double bond. This is a omega-6 because the first double bond is detected in the 6th carbon. 1, 2, 3, 4, 5, 6. All right. So you will have arachidonic acid. You will hear it very often later on in my presentation. This is a long chain profile which has 20 carbon. It has 4 double bonds. Okay. And the last one is a very long chain fatty acid as well. This is 20 carbons and it has 5 double bonds. All right. So let me just go out from this slide. So you need to remember their names. These are the prominent ones. Okay. So let me just go to the next one. Well, for PUFAs, we have actually 2 main naming system. What we call, or the other word is nomenclature of fatty acids. So we have the omega nomenclature. We can have the delta nomenclature. I will start from the left, which is the omega nomenclature. So it carries the omega sign, all right? So I will take, I think the easiest one is linoleic acid because it has fewer double bonds. So For linoleic acid, when we start counting using the omega nomenclature, so we start from the methyl N here. It's mentioned over here, methyl N. So we know this is like 6, omega 6, because the first double bond is detected at the 6th carbon. So it's 1, 2, 3, 4, 5, this is 6. And then we put a double bond separated by another carbon. And then because it has 2 double bond as indicated over here, so there's another one that is detected, where is it? At the 9 position. 6, 7, 8, 9. Yeah. So when we report under the omega nomenclature, is written this way. So I think you've been told from the very beginning, this is very easy to follow, isn't it? And for delta nomenclature wise, like it starts from the carboxyl and the carboxylic acid here. So this is the first carbon. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9. Because half of 18 is 9. All right. No, I shouldn't say it here, but maybe it applies here better. Anyway, so you can see 9 here. You can see 9 here as well. Same here. Let me just move it out. Okay. So then the next one is, you ignore this, maybe I just erase it better. That's my eraser. Okay, so I have a blue color, so I should use red instead. Anyway, you get my point. So this is 9, this is 10, okay? This is 11, this is 12. Okay, so for delta nomenclature, it requires us to report all the position of double bonds. That's why you see this, okay, 9 and 12, because 9, 10, 11, 12. Okay, then you get another double bond here. So that's why you have, when you report it under the delta nomenclature, you have 18, 2, followed by the delta sign, which is a triangle, 9, 12. Then you go for linoleic acid. Okay, so you need to write more. Let's say for EPA. For EPA, if we follow the omega nomenclature, this time I got it right in blue. Okay, so we start from the methyl N because it's omega 3. So you have 1, 2, 3, right? So the first double bond is detected in the 3rd carbon position. So that's why you write like this. And because it has 5 double bonds, so therefore it's 25. It's very simple, very straightforward, isn't it? You need to be a little bit hardworking for delta system because you need to declare all the double bond position from the carboxylic end. So this is 1, okay, 2, 3, 4, 5, okay. Then you have a kink every time you have a double bond like this. So because there's 5 double bonds, so you have to report one by one. 5, 8, 11, 14, 17. So for your information, nowadays, like even in food science and food technology, the omega nomenclature is used more often compared to the delta nomenclature. But still, the delta nomenclature is still used nowadays, actually, when we want to talk about the enzymes and so forth. You will see that in a while, okay? So based on your preference, may I ask, like, which nomenclature do you prefer, the omega or the delta nomenclature? If you like omega, you can type the omega. If you prefer delta, you type delta. I'm just doing a quick survey. Now we have 3 students respond to me: Tibian, Nosuraya, and Chong Jai. Three of them, they prefer the omega nomenclature. Now, I don't know about the rest of you. Are you still listening? Anyway, I'll have more questions coming up, okay? So there you go, the nomenclature of, there are two deltas, okay, coming up. Anyway, so there you go, the omega nomenclature and delta nomenclature is to help you to understand what I'm going to talk about later on. So now you know there are different types of fatty acids. You have the short chain, you have the MUFA, which is monounsaturated. You also have the saturated fatty acids, you have the PUFAs. So now I think it's also a good idea, since we talk about them, that we can have a look at the functional properties of these lipids in general in food processing, because we consume them every single day, right? So these are the factors that determine the functionality of fats and oils, okay? It will determine your choice, okay? When it comes to like when you want to apply, you want to select or choose the types of oil that you want, especially for those who want to become an entrepreneur, or you have a factories, or you want to cook, or you want to bake, okay? That concerns a lot. Okay, so by knowing which type of fats and oils that you're going after, it will affect the result of your product. Okay, so there are actually four main factors that I want to highlight here. The first one is melting point and melting profile. I'll just stay general, okay, for this talk. So like we know that a lot of the times, like palm fat has a very wide application, but all due to its a wide spectrum of melting temperature. It can go low, it can go high, depends on what kind of fractions we are talking about. Okay, so for instance, we can use palm fat and incorporate that in the emulsion of burger or patty, okay? So this will actually, because of the application is in burger or patty, we need to cook it. So we need to select palm fat that melts at high temperature. So I think it's above 100 or above at least 70, okay? So this is palm fat, all right? Conversely, we have another fat that we know very well, which is cocoa butter, and it is used in, guess what? Chocolate making, isn't it? So cocoa butter has a relatively low melting point compared to palm fat. So it melts even before it gets cooked. So that's why when we have cocoa butter incorporate in our who likes chocolate? Who doesn't like chocolate? Okay, so when it's incorporated into chocolate, actually it gives you the melt in your mouth sensation. Compared to palm fat, right? It will get waxy if you swallow your chocolate, if the formulation has a lot of palm fat in it. I mean, I'm talking about chocolate, okay? So the melting point of the fats and oil of your choice actually will determine how you feel when especially when it's incorporated in food, all right? The second one is crystallization characteristics. We know actually fatty acids, they can crystallize, okay, at different temperatures. So this is especially very important when it comes to chocolate making, because I think this is the best example that I can get so that you can understand what I'm talking about. I think some of you have the experience of melting chocolate and preparing it as a coating for cookies or even cake, maybe ganache, okay? So melting chocolate, it takes a great precision in terms of controlling the temperature, because the amount of cocoa butter and whatever fat that you put in actually will affect the appearance, and also what else? The appearance and the liquidity of the chocolate if you use it as a ganache or coating. Okay, for making chocolate bar, it also has the same more or less the same effect, because if you melt it at very high temperature, the fat crystals actually they will form a different type of crystals, and it will make the chocolate feel looks a little bit dull. Okay, and it does not give you the snap that you want. Okay, if you're making chocolate bar, it will become like soft. So if chocolate is melt, okay, at a temperature, let's say instead of overheating it, like let's say if you control around like 30 degree or 31 degree, which we call it tempering, okay, with proper tempering, actually we can get a chocolate that is shiny, and it gives you a good snap when you break it, because the crystal that is forming in the chocolate, it actually can form, it will be well distributed, and then the size of the crystal will be just right. I would say like it has a certain size that we need to meet, but for the sake of this talk, I'll just try to simplify it that the crystal will be well distributed and the size is relatively maybe small to say, okay? And let's move on to the third factor, which is storage stability. As we all know now, like there are different types of fatty acids, which we have the short chain fatty acid, we have PUFA, and we also have saturated fatty acids. So different types of fatty acids will give you different stability. The one which is most susceptible to oxidation is actually, anyone knows which one? SCFA, MUFA, or PUFA, or SFA? It's okay, I'll answer it myself. The one that is most susceptible, means that is prone to oxidation, is actually those which has unsaturated bond, especially this one is the most susceptible to oxidation, not so stable, because the double bond will tend to, I'll put it in a very common word like break, okay? So it's prone to oxidative and also hydrolytic processes. So the choice of our oil, we also need to be careful, because at the end it will lead to, if it is in food, we can see, we can feel actually the food becomes rancid. It has a smell. In Malay, we call it dengek. Okay. In Indonesia, what do you describe it as? Any word for that? Rancid. Rancid. It's okay. We can look up the dictionary. Okay. So let's move on to the fourth factor, nutritional requirements. Well, as we all know, like in different stage of growth, okay, from let's say from infants to teenagers to adults, even to like what do you call this, midlife, or the elderly, different stage of growth, we have different requirements in terms of nutrient intake. Okay, thank you very much, Dengek. Thanks, Ms. Shabina. So for nutritional requirements wise, like we can see in the market, like there are different recommendations of fat or even omega-3 and 6 intake recommended. on the food labels, especially in functional foods. So because of this, like a lot of manufacturers, food manufacturers, for instance, they try to, how to say, they try to meet and they try to claim, okay, what is good about their product. But of course, these claims has to be verified by the respective authorities. In Malaysia, we have Kementerian Kesihatan Malaysia, which is the Ministry of Health. And what about in Indonesia? Are you referring to your FDA, or you have your own special name for your Kementerian Kesihatan Malaysia, the Ministry of Health? It's also known as Ministry of Health.
Yeah, in Indonesia, I think Menteri Kesehatan, if I'm not mistaken. The same was.
Yes, okay. Good to know. Thank you very much, Ms. Shabina. So there you go. The 4 factors that will determine the functionality of fats and oils. It depends on how we select them. So let's look at the functionality of fats and oils. So I'm not going to go through all of them, but I'll just pick a few of them so that you can have an idea. So for instance, like fats and oils are used to, let's say, to give solubility. We all know that there are certain food ingredients, for example, because I'm a food person. So we have like fat soluble vitamins, we have fat soluble flavors, okay? So actually fat can dissolve these, like what I mentioned, fat soluble vitamins, they include vitamin A, D, E, K, right? So you can actually use fat as a carrier to dissolve your vitamin A, D, K, or whatever flavor that you wish to incorporate in your food product. So for example, milk fat can dissolve fat-soluble flavors and vitamins in the product, okay? So you can actually like enrich your milk, let's say, with vitamin D or E, for instance, all right? The next one I want to pick is moistening ability. Moistening ability, it says what is the mechanism, because fat prevents dough or batter from drying out after baking, actually. So therefore, at the end, as a result, your cake will feel moist, okay, after baking, because it does not dry out your product, all right? And lastly, we use oil as a medium to transfer heat, because the temperature actually can go up till like, or even higher than 170, I think, yeah? So it is a very good heat conductor, and also it will make the water vaporize. So it will give a very crunchy and also crispy food texture. Mainly this one we say is for frying, okay? So a lot of people like fried stuff because it's crispy, right, and crunchy. So for extra information, you can actually have a look at certain journals for extra reading. It's over here. So if you're interested, you can take a snapshot and read about this later on. So far, so good. If you're okay, give me a thumbs up. If there's an emoji for that, let me go and find it. I see smiley face, but yes, there is actually a thumbs up. Here we go, my thumbs up. So if you're doing good so far, give me a thumbs up. We are about to start with omega-3 and omega-6 fatty acid synthesis. Okay. So here you go. Well, to simplify things, okay, I'll try to make it as simple and as possible by using I would say like simple phrases, okay? So Remember earlier on, I said linoleic acid is actually essential fatty acid because our body cannot make it because we are lack of two enzymes. We cannot make linoleic acid. We cannot make alpha linolenic acid in our body because we are lacking the delta 12 and delta 15 desaturase enzyme in our body. All right. So this delta 15, I think it coincides with the omega-3, and this one is omega-6. All right. So when we want to get linoleic acid, actually we can get it from a lot of plants. Okay, plant sources such as soybean oil, palm oil. Yes, we can have some amount of it. We can get it from seeds and nuts, okay, from a lot of plant sources. Okay, and actually this whole bar is blocking my view. I believe it is like this is delta 15 desaturase on top. Okay, there's a word there that is covered by this thing. So this is exclusively found in plants only, the enzyme. So this enzyme actually can convert linoleic acid to alpha linolenic acid, which is a omega-3 fatty acid. So from omega-6 convert to omega-3 by using delta 15 desaturase. Okay. Okay. So let's look at on our left. We have the synthesis, the biosynthesis of omega-6 fatty acids. So here, when you have linoleic acid comes in, then our body has certain enzymes. We have the delta 6 desaturase, we have the delta 5 desaturase, we also have elongase. As the name suggests, desaturase actually will add on double bond. Elongase will actually add 2 carbons to make the chain, the hydrocarbon chain, longer. So as you can see, whenever there's a desaturase that comes in place, there is an addition of double bond over here. Easy to follow, right? Whenever there's elongase that comes to place, we have actually change of chain length +2. Okay, so same goes to here. You have the desaturase, so you will have addition double bond over here. So the flow goes like this. You have linoleic acid that it will be converted to gamma linolenic acid. All the way they are omega-6. Okay, and then you have the diholol gamma linolenic acid, and in the end, most of the time, you will have arachidonic acid. All right, so this is the flow. The main things I've already highlighted here so that you can grab the important ones. You have the linoleic acid as the starting point. You have the arachidonic acid as the end product. All right. Let's move on to the other side. We have ALA, alpha linolenic acid, to start with. So this, you only get it from plants because we cannot make it ourselves. So once ingested, actually our body has these enzymes, okay, similarly, like what I explained to you earlier on. And it will go through a series of biosynthesis to make longer PUFAs. They are the eicosapentaenoic acid, okay, the eicosapentaenoic acid, and at the end you have DHA. Let me go through one by one because some of the names will appear again later on in my slide. For alpha linolenic acid, Whenever you have a desaturate, that means you will have addition of double bonds, right? There you go, you have this stearidonic acid, all right? And this one, I don't put the name because I didn't go and find it, but I would say like, if I have time, maybe I can identify, but at the moment you can remember it is like 24 and 3. And then with this, is you can have your eicosapentaenoic acid, which is a very like well-known fatty acid in most of the functional food. Okay, so by adding one double bond, then you get your EPA, and it tends to get longer, you get your DPA because it's 22, all right, with elongates. It will further elongate, but later on it will go to beta oxidation. Okay, then the 2 carbon over here will be taken out. Then you can get your docosahexaenoic acid, which is 22,6. I want to highlight that the whole synthesis process, okay, you can see there's a lot of steps involved, right? So I would say in biosynthesis, there's no 100% efficiency. So because we have a lot of hurdles, and sometimes like mitochondria, based on my reading, they also need like, because they are the powerhouse of our cells, they will also burn energy. So sometimes fatty acids will be utilized to obtain energy. So the whole process actually is not, that's why it's not 100% efficient. So that's why, like, although our body can synthesize EPA, DHA, and DPA in our body, but we cannot meet our demand, okay, because of this whole biosynthesis is not like a very efficient, okay? There's limitation to put it in a very simple form, okay? So that's why there is a need to obtain all these omega-6 and omega-3 fatty acids through our diet. We are not just focusing on what? LA and ALA alone, okay? Somebody's okay. So let me get a magnifier. So on the left again, we have omega-6 fatty acids. So the main sources of omega-6 fatty acids, okay, because we cannot make linoleic acid ourselves, as I mentioned, we can get it from a lot of vegetable oils because most of the time we get vegetable oils from seeds. And in most cases, seeds actually is the reservoir of fats and oils, okay? For example, sunflower oil and so forth, okay? So we take the oil and then our body will convert it. Again, it's not 100% efficient, all right? We can also get arachidonic acid through our diet, such as meat, poultry, and eggs, all right? Let's look at on the right. Now we are talking about omega-3 fatty acids, okay? For ALA, which is alpha linolenic acid, because our body cannot make it, thus we call it an essential fatty acid. We can actually get ALA from a lot of plants, especially green leaf vegetables, okay? As well as seeds and nuts, such as flaxseed, chia seeds, all kinds of nuts, okay, or cereals, all right? And later on, it will be converted to all these long chain profiles, such as EPA, DPA, and DHA. Because of it's not 100% efficient, when we biosynthesize it, we can also get EPA, DHA from fish, mainly, and not just from fish. We can also get these from algae oil or krill oil. I will talk about krill oil in a while. If you don't know what krill is, anyone can guess what krill is? Krill, Krill. I think if you move your fingers a little bit and Google it, I think you will see what kind of creature is krill. Now, a little bit of someone says krill is a type of fish. Nice try. Close enough, but it's not a fish though. Now back to my slide. A little bit of introduction on the health benefits of polyunsaturated fatty acids, health benefits of PUFAs, because why people are talking about EPA, DHA, maybe even arachidonic acids, okay? So all these PUFAs actually, they have different types of health benefits, mainly for blood clotting, because they are involved in platelet aggregation. And some of the PUFAs actually, they are pro-inflammatory because they are used to produce cytokines. Well, I just want to say inflammation is not a bad thing because it's actually a normal occurrence of, let's say, cell renewal. It will send out signals to repair or to replace cells that are old or damaged, okay? But excessive inflammation is a bad thing, all right? I just want to stress. And in PUFAs, we also have anti-inflammatory metabolites. I mean, like PUFAs can actually affect the production of anti-inflammatory metabolites. Okay, so actually there are two sides of the coins. You have the pro-inflammatory. You can also have the anti-inflammatory. I'll tell you in a while, okay? And also PUFAs also involved in neurotransmitter because they are involved in the induction of release of acetylcholine. Yeah. So other than that, scientists also pointed out that PUFA-rich diets also can help you in terms of immune function, blood pressure, cholesterol, and triglyceride levels. Maybe they will change your profiles, okay? And also, it will affect your cardiovascular function, okay? So I have an example when there was, so this study was carried out in year 2014 by supplementing EPA and DHA at 3.4 grams to patients with heart problems. So their blood profiles are taken before the surgery and after surgery. So supplementation of EPA and DHA actually is said improve the metabolic and anti-inflammatory, say that again, improve metabolic and anti-inflammatory antioxidant capacity of the heart. But it's because when they measure, they have done a comparison on the blood profile, they notice that there's a reduction, significant reduction in omega-6 fatty acid, and there's an increment of omega-3 fatty acids, especially the EPA. All right, this is a potent, I would say, like antioxidant. Okay, so that's why they say, like when they have done some tests, they say like the myocardial tissues, the heart tissues taken from the treated patients, they have actually better antioxidant capacity, and also there's expression of anti-inflammatory enzymes. what I'm trying to say is that for people with certain needs, let's say like health, state of health, like depends on the doctor's recommendation. In this case, it did help, okay, patients with heart problems, okay. You can see there's a change of a profile and also expression of certain enzymes in their heart tissues. Well, not just that, for PUFAs, it also has a connection with gut microbiota. Like what I say just now, not just the short chain fatty acids, PUFAs also has a role to play in our gut together with gut microbiota. Because like I said, in our body, we have a lot of strains of different bacteria. So people started to pay attention on the probiotics, especially the lactobacillus strains. Okay, these are the strains. So they noticed that when they supplement PUFAs, it could be the C18 ones or so forth. And when they supplement these PUFAs to the probiotics, they can notice that there's a change, okay, of the fatty acids profile, especially the EPA, okay. So another example is when fish, they are fed with PUFA diets, okay, they can, people detected that they are more LAB, lactic acid bacteria, which are probiotics, okay, are found in the GI tract, the gastrointestinal tract of the fish. This is a very good indication because like it suggests that it could be an alternative to drug usage and hormone usage. It can, I would say, maybe it can reduce because nowadays people are moving to a more so-called natural way, okay, to how to say, to raise animals, okay, as food, right? So now that you have learned what is omega-3 and what is omega-6, all right? So what is the ideal intake of omega-3 and omega-6 ratio? Because there is a ratio involved, all right? So here, this is a diagram, and it is actually a triangle. From the left, we can see there's a green zone here, and then the color started to change from green to yellow to orange to red, right? And then we can see, like it says, like high omega-3 diet, and in the middle, we have low omega-3 diet, and we have this Western diet, omega-6 diet. Excuse me. I take a sip of water. And at the bottom, you have a scale that runs from 1 to 15, sorry, 50. That is for omega-6 to omega-3 ratio. It means simply say like this is like something like this is a nominator, this is a where is it? Nominator, denominator. So here, this is in the green zone, it's actually like a high omega diet, so it's 1 to 1. If we go here, this is like almost like what? If we go all the way here, that means it's 50 to 1. Okay, I hope you can follow. So for the green zone, high omega-3 diet, I'm not so sure if anyone can achieve it, okay? Because maybe during the old days, like when humans are hunters, are gatherers, there is a possibility to achieve it. Because like we hunt and we gather nuts, fruits, okay, from the wild, all right? There is a possibility. Or to make it more relevant to us, do you know there's a population called Eskimo? or they are known as Inuit. So actually they're living on the North Pole, right? They are scattered all over in the North Pole, and they hunt, let's say, seals, whales. So they eat a lot of fish all year round. Okay, then that's their main diet. They could be falling in this particular zone because their diet is rich in omega-3, all right? But as modern humans, because our diet consists of mainly nuts, corn-based, grain-based materials, okay? I will show you in a while, okay? So therefore, we might be falling within this zone, or even worse, in this zone. Can anybody tell me what is Western diet to you? Because it's labeled as Western diet over here. What do you think? What can you describe about Western diet? Anyone? What do you know about Western diet? Someone says consume more fatty food. Someone says fast food. Anyone else? I can hope to get more response in the chat box. More green, Western diet, okay. Any more idea? A lot of meat, maybe. Dairy products, someone say, I would say, well, yeah, we can consider. Anyone else? This is an open-ended question, so there's no right or wrong. So feel free to, to try. Actually, I'm trying to show you a page, but it's not working. I don't know why. So let me just take a little bit of time. So keep the answers coming. So what do you think? What is Western diet to you? High processed food and meat. Yeah, okay. That's also in my mind as well. Good. A very good response, I would say. Let me share the screen. This is a screen taken from Wikipedia. So if the screen is visible to you, please type 1. 1. Number 1, if you see it. Okay, according to Wikipedia, thank you very much. According to Wikipedia, it says Western diet pattern is actually like things that are high intake in pre-packaged food. I would say it's processed, highly processed food, okay, convenience food. Refined grains. Refined grains, it could be like white bread, let's say, all right, or cookies, okay, refined grain flours, red meat. What is this? It's like a plate of beef, okay, and processed meat, high sugar drinks, a lot of sodas or soft drinks, candy, sweets, fried foods, conventionally raised animal products, butter, other high-fat dairy products. Okay, some students got it right. Eggs, potatoes, corn. Corn, you can make a lot of stuff out of it, including high fructose corn syrup. And then what else? Low intakes of fruits. You don't have to get the picture, I guess. Vegetables, grains, pasture-raised animals that are free running, I think. And fish, nuts, and seeds, these are low, okay? So this is according to Wikipedia, how they define Western pattern. But to me, I would just feel weird if they point the finger to Western diet, because you have salads, right? You have like, I would say like soup, that sounds very healthy to me, okay? A lot of veggies and potatoes and so forth. But this is how people define Western diet or Western pattern diet. What I want to say is that like foods that are highly processed, I would say mainly with deep fry, I would say. Some students say fast food. I think I can agree with that, okay? But as we are now in the era of globalization, I think like maybe it's not that appropriate to like sort of like accuse, okay, to point fingers to Western diet as the main contributor to our high omega-6 diet. Now let me just go back to my Zoom and try to find the share button. Now where's my share button? Okay, coming back to my screen. All right. So now you know, sort of like try to differentiate, okay, the diets, okay, that will lead to different ratio of omega-6 and omega-3, all right? So according to Katayama, okay, the best ratio, if we can maintain, is 4:1. That is actually 4 parts of omega-6 and 1 part of omega-3. That's the best. I don't think we can go to 1:1. It's a bit tough, I would say. And according to a lot of scientists, this is a generalized statement, okay? They recommend like omega-3 long-chain PUFA, those like from C2022, trying to get about like 250 mg, which is equivalent to like 2.25 grams, okay, per day. And there's a association of the global, I can't remember what it's O stand for, global something. That's actually a association that is focusing on EPA and DHA. They recommend a daily intake of 500 mg, which is 0.5 grams of EPA plus DHA, okay, per day. So this is the recommended intake of this is omega-3 actually, so that you can know like how much omega-6 you want to take. All right, so how come we can get such a high ratio of omega-6 in our diet? Like most of us, I don't know, we could be falling within this zone, okay? So let's look at some examples of food. I'll take chicken, for example. The conventional way of raising chicken, we can see the fatty acid profile. If we divide it into omega-6 and omega-3, actually we can see there's a very high proportion of omega-6. Yellow is actually omega-6, yeah, compared to omega-3 in the chicken meat. All right. And the free range has slightly lower, which is 19:1. Pasteurized means maybe they can run freely around the field, okay, because it's a green pasture. And free range doesn't mean that they are they are running in the field. Free range can be they are free running in a farm with cement floor, who knows. But anyway, the way we breed animals, okay, the food that we give to animals actually can have a very direct impact on our diet. That's what I want to say. Another good example is the beef, okay? So cows that are grass-fed, when compared their omega-6 to omega-3, is actually 1.7 to 1, okay? When it's fed with corn, which is a very common ingredient in animal feed, okay? Corn, jagung, is actually 17.7 per 1. I'm talking about omega-6 over omega-3, all right? So it's 10 times difference, grass versus corn, all right? So that is a huge difference. So that's why what we give to animals, like I said, it will impact our omega-6 and omega-3 ratio. So let's look at, again, the biosynthesis of omega-3 and omega-6. Why is it so bad if we have high omega-6 in our body? So I will use this particular flow chart to explain to you. Remember, on the left, we have omega-6. And as we go through with the biosynthesis flow, okay, actually, if you notice that they are actually metabolites, which we call cyclooxygenase and lipoxygenase. Actually, they are the metabolites that will regulate our inflammation reaction in our body. So the production of series 1 and series 3 metabolites, okay, actually they are anti-inflammatory. Okay, done. But as we take more omega-6 fatty acids in our body, let's say we keep on ingesting linoleic acid, or we take more of this, it will lead us to produce more arachidonic acid. And in the end, it will give rise to series 4 and series 2 metabolites. These are like it could be leukotrienes, it could be lipoxin, it could be prostaglandins, it could be thromboxanes. These are, remember, these are series 2 and series 4. They are actually pro-inflammatory. All right. It's very different for the synthesis pathway of omega-3 fatty acids. If we go down the pathway, eventually this long chain PUFA actually can give rise to series 3, series 5 metabolites, which are anti-inflammatory. Okay, so if we have too much metabolites that can govern, that will lead to inflammation, that is bad for our health. All right, because a lot of chronic diseases is started with long-term or chronic inflammation. All right, so let me sum it up again. So like how does, in other words, how does omega-6 and omega-3 actually can affect inflammation? So actually omega-3 and omega-6, okay, how do they act is that you know that omega-6 PUFA can lead to pro-inflammation because they will produce metabolites that are pro-inflammation. So omega-6 and omega-3 PUFAs will come in and as a to compete with, like in terms of substrate competition. So they will produce a different set of metabolites, okay? So it will compete, at the end, you will have less pro-inflammatory metabolites, all right? So these will actually reduce the inflammation, or they can also be converted to some other metabolites that are known as resolvins, protactins, and myrcins. They are actually anti-inflammatory. So the rest of the slide, I think I will not go through it due to time constraint because I still have a few more slides to cover. So for omega PUFAs, in summary, like actually has the ability to give you anti-inflammatory effects. Okay, it also have like a certain types of role for example, in giving a therapeutic effect for those who are undergoing chemotherapy, because it can improve the efficiency and tolerability of chemotherapy in patients. Okay, it is still in study though. Excuse me. So let's do a little bit of practice. I have a table with fatty acid composition. They are normalized to percentage. This is not the actual amount, but excuse me, but the percentage of saturated fatty acid, mono unsaturated fatty acid, and polyunsaturated fatty acids are listed according to the columns, and as well as the total omega-3 fatty acids and total omega-6 fatty acids are listed in these columns. And at the end, you have the omega-6 to omega-3 ratio listed in this particular column. So my question to you is, if you were to choose among, let's say, I need to draw a line here. Okay, among the plants, so what kind of oil are you going after? If you want to maintain a ratio of omega, if you want to achieve omega-6 4:1, let's say. So what is your best bet? May I see some answers from chat? Among the plant-based oil, pick one: sunflower, corn, soya, palm, or olive. It looks like very quiet here. Somebody give me a soya with a question mark. I need to be confident. Anyone else other than Li Shiqi? Come on. Tivia said olive. No, Soraya, not yet fish, not yet going to fish. Look at here. The first box number 1. Sunflower, corn, soya, palm, olive. Soraya says corn. Anyone else? Sammy Jungji says palm. Anyone else? Keep your answers coming. By the way, MC, can I do a time check with you? Am I running out of time? This would be my last few slides. Miss Shabina, how many minutes more do I have?
Yes, that's
How many minutes do I have?
it should be till 10 o'clock, but it's okay. You can just I can give you like another 10 minutes maybe.
Okay. I think this is my last few slides.
All right.
Okay, so kind of short, actually, your choice of oil, I would recommend you to stay away from sunflower in this case, okay? Because the ratio of omega-6 is crazily high, I would say, based on the number, all right? 652, it means like this, all right? So if we go after the fatty fish, these are all fatty fish, all right? So they are very good choice because like this is actually like a 4/100, isn't it? All right. So for microalgae is also a very good choice as well because it gives you the ratio is like this. So we are aiming actually the omega-3, which is the bottom number. We try to make it bigger. Okay. So aside from that, we can also have a look at types of oil that we use in our daily cooking. So this is another normalized fatty acid content. So this is not the actual amount again, but it's separated according to the saturated fat, monounsaturated fat, and polyunsaturated fat. As you all can see, like the one that is omega-3 is actually, omega-3 is actually yellow and omega-6 is actually orange. So the one that is highest is actually flaxseed oil, right? And or if you are just after the PUFA, actually like you can find a lot of the plant oil actually that are relatively high in PUFA. Okay. And I just want to stress for olive oil has a very high proportion of oleic acid, which is monounsaturated fat. This is also a very good oil for cooking as well, because it has a high oleic acid content. I just want to stress for oils that are high in saturated fat, which is in blue, we try to make it like take it in moderation. Because doctors normally don't recommend you to take a very high proportion of this fat in your diet, all right? So that's why since we know PUFAs are so important in our diet, so a lot of food manufacturers, they are making effort to incorporate EPA and DHA, especially those that are extracted from fish or microalgae. All right, in our food products. This can be observed. These are the examples of food products that we can get in our Malaysian markets. There's one issue in fish oil because it normally gives fishy odor, but there are technologies that can actually eliminate the volatiles, the unpleasant volatiles by doing very extensive purification. Then it's actually making it possible to incorporate into food products such as dairy, like milk powder and so forth. All right. So in this particular slide, I just want to give you, it's just like a reference to you all. This is a journal that I extracted from the Malaysian Journal of Nutrition. It's actually done by a doctor. He actually took the typical diet of Malaysian and try to compare the intake according to the recommended nutrient intake in Malaysia versus what is suggested by the World Health Organization. It seems that like a Malaysian's recommended nutrient intake in omega-3 fatty acids is way too low compared to what is suggested by the WHO. So what I can recommend is that if we can't have like the access to perilla oil or flaxseed oil, which is relatively high in terms of omega-3, we can actually go for a combination of different food sources in order to maximize the omega-3 fatty acids in our diet. This is achievable, all right? So in conclusion, for PUFAs, We know that actually there's enzymatic oxidation involved when we have PUFA because they are susceptible to oxidation. In our body, when we have excessive omega-6, it is pro-inflammatory. But if we have omega-3, actually it can help us to anti the inflammation. All right. Outside our body, like especially in food products that are very high in fat contents, actually they are prone to autooxidation and photooxidation that can create different harmful substances such as alkanes, aldehydes. If accumulated at low dose, it's still okay, but when it's high dose, it will induce oxidative stress and inflammation, eventually can affect the product of the food product by giving a off odor, such as rancid. And as for the efforts of biotechnology or food biotechnology, actually there are many efforts done to find alternatives rather than depending on the traditional fish oil. So here we have krill. This is actually a crustacean, okay? It can be as small as this, it can grow as big as maybe a few inches, okay? Krills are normally found in the Antarctic area or cold weather area. So this is their scientific name, Euphrasia superba. And it is actually, the krill oil is actually generally regarded as safe, okay, by the US FDA. And is regarded as novel food in the European Union. Well, krill oil is very much promoted because it's rich in omega-3 PUFAs, especially EPA and DHA, and these EPA and DHA are mainly bound in phospholipid forms. It's said that phospholipids actually, this kind of EPA in phospholipid bonds are actually, they are better absorbed, more bioavailability than the fish oils which are exist in triglyceride forms, okay? So this is a typical picture of krill oil product. Why is red? One of the reasons is that it has the krills actually contain astaxanthin, which is a red pigment, a potent antioxidant. And another way of looking at it is some manufacturers, they colored them because in nature, like everything has its own, because the krill that we harvest may have a different concentration of pigment from season to season. So for aesthetics reasons, so they colored it red as well. All right. So krill production. So normally krill is harvested from the sea because krill is susceptible to degradation. They are very sensitive. The lipids will degrade very fast. So it has to be processed. So therefore, people have a ship for production. It will be cooked right away upon harvest and pressed to get the meals. So the meals will become in solid and it will be sold as animal feed later on. This is the dry product. For the liquid, after pressing, it will be separated by a separator where you get water phase and crude krill oil, which you need to be refined later on. Okay, so for refinery, you can have the phospholipid and also the other parts of the krill oil in different fraction. Okay, for genetic modification wise, biotech has a lot of like efforts done. So because we know that the biosynthesis of PUFA, especially the long chain ones, is not 100% efficient. So we try to improve by genetically modified plants. Okay, so plants that are genetically modified, such as flaxseed, Camelina sativa, actually they can make EPA and DHA in the seeds. All right, so very high proportion though, I can say, because it's 23.3 of the total fatty acids in the seeds. And there's also another flower called Brassica carinata. It's also have very high EPA, and there's also transgenic mustard from BSF, and also transgenic soybean. All these efforts are to raise the long chain profile, especially the EPA and DHA. So the traditional flaxseed that we see, actually, it has half of the total omega-3 is actually, like half of the total fat contains omega-3. So EPA and DH for GM, after GM, like genetically modify another half of the total omega-3 is actually consists of EPA and DHA. All right, so this is a very good improvement. For soybeans, another company, Monsanto, it came up with the idea by skipping ALA in the synthesis. So instead, they enrich the stearidonic acid, which is one of the fatty acids in the pathway of the synthesis of long chain PUFA. So it will reduce the hurdle, okay, so that it can increase more EPA and DHA in the soybeans. It is done by inserting two genes from a flower and a fungus in the DNA of soybean. Okay, so for further reading, you can have a look at these journals because I find that they are very informative in terms of knowing better about the health benefits of omega-3, as well as utilization of PUFA in food preparation and animal feed. So that's all my presentation. So this is my conclusion slide. I'll just read it out very fast. So fatty acids, actually, we have different distribution in animals and plants. We know that omega-6 PUFA that leads to very high omega-6 and 3 ratio. It can actually promote development of chronic diseases, but when we have very sufficient or increased amount of omega-3 PUFA, it can suppress these effects. We need to know more studies about all these fatty acids though, and we can actually manipulate the distribution of omega-3 and omega-6 fatty acids in plants and animals to achieve good health. Okay, I think that's all for me. Thank you very much, and sorry for taking too much time. Okay.
All right, thank you very much, Dr. Pui Khoon. If I pronounce it right.
Yeah, it's correct. Pui Khoon.
Pui Khoon. All right, so Doctor, would you like to have this Kahoot quiz, right?
I'm not so sure because I think due to time constraint, I'll just go with the flow from the organizer. So because I have taken too much time.
Okay, I think maybe if there's like one or two questions to Dr. Hong Pui Khoon, maybe, yeah, before we move on to the next session. Any questions from the floor? I think this today topic is a bit heavy maybe for certain students because but it's okay, it's quite insightful as well. So lots of students can learn. Okay, from Shikili, will the omega-3 will be destroyed by heat?
Yeah, depends on the temperature. That's why PUFAs, they are very susceptible to oxidation and heat as well. I forgot to mention. So a lot of the times, especially when we buy supplements, they will tell us to store at room temperature or very, very cold place. I think that's one of the reasons to preserve the potency of omega-3. So yeah, we still need to cook food, but if we cook it in the right way, we still can preserve some omega-3 fatty acids in food as well. Did I answer your question?
Okay. Does it answer your question? Okay. Any more questions from dear students? Doctor, I want to ask one question. Maybe it's a silly one, but what do you think about the current technology, which is the fry cooker? What it's called in Malaysia? The frying cooker?
Air fryer, you
Air
think?
fryer, yes. And because like it's not going to use any oil or something like that to avoid the use of like palm oil. What is in your opinion on that?
Well, everything has its two-side story. Well, there are pros and cons though. Like for me, I seldom use air fryer. Uh, well, how to do it? Well, sometimes food will taste a little bit like less without the oil. That's one thing. But with what so-called like healthy eating, for me is very subjective to certain people. Okay, but for me, air fryer, I see it is if you use it at a lower temperature at a controlled manner, and actually you can achieve like the purpose of like using less oil in cooking. I think that's the intention of most people when it comes to using air fryer, right? But when you fry it up at a very high temperature, you might as well like if you use very little oil, but sometimes like when you use the air fryer, if you do use a very high setting, we'll also see like some foods, it will not turn out the way you want it to be, like excessive browning in the food if you experience it. So this browning, if you take it like very often, like brown food, I think is for me, I think you need to be practice more what's the word, moderation, because like it's a mild reaction product. It could be good, it could be bad, but if excessive, it might also give you some detrimental health effect as well. So there are two sides of the coin. So I would say you need to practice in moderation. Air fryers is good invention, but we try to use it as according to what is recommended by the manual. Yeah, we don't abuse the temperature.
Thank you for your answer, Doctor. Okay, any more questions from the students? So in Malaysia, there's so many Creole fish, isn't it? Or no?
Well, actually, the big companies, they are coming from Norway, Japan, US. So they are the main players in this krill oil industry. So most of the time I can see the krill oils that we consume, actually they are imported.
It looks like people call it like a shrimp, right?
It looks like a shrimp, but it's a different species.
Okay, question from Effie Fever. Hello, doctor. What is the difference between normal olive oil and extra virgin olive oil?
Well, based on my knowledge, I think it's between the fraction that is obtained and also the process that people went through to get the oil. As I understand, like extra virgin olive oil, correct me if I'm wrong, because I'm not in that industry. According to my understanding, is that like people normally do it cold press, so that's why they try to get that's the first fraction that they get. So it has a very high polyphenol content together with the olive oil. Whereas the normal olive oil, it would have maybe relatively less polyphenol. So the ones that is extra virgin olive oil, because it has a different fraction of oil composition, okay? So it's recommended to use it for, let's say, for salad dressing or maybe just dipping. And for the normal olive oil, because of the way it's processed is different, it's coming from a different fraction of the oil. So I would say probably the it can withstand higher temperature. So that's why people will recommend that to be used in cooking, let's say saute and so forth.
All right, any more questions?
Are we killing time? If we have time, why don't we just go for Kahoot? But my Kahoot would take like, I would say 10 to 15 minutes if time permits. If not, then I'll just go with the flow from the organizer.
Okay, it's quite long, 10 to
Yeah.
15 minutes. All right, if there's no more question, maybe we could take a quick break before we continue with the second session. And before that, maybe from Dr. Pui Khoon, if there's any words for the students before we end our first session.
I just want to say, yeah, today's topic might be a little bit heavy for you all, but I just try to introduce to you this topic because it could be an interesting one. Hope you learned something new today. And apologize for the time spent. I think it's a little bit way too long. Okay.
All right, no worries, doctor. It's actually a very interesting topic, and all of the students could learn something new from this, because normally we just learn about saturated, monosaturated, but not into a more detailed in this omega-3, omega-6. So yeah, so the students can gain some of this knowledge from you. So thank you very much, Dr. Hong Pui Khoon, for your time and a really insightful topic for today. And we hope maybe another day can see you again. All right.
Okay.
Okay, thank you very much, Doctor. And for dear students, we'll take 5 minutes break and then we will continue with the second session directly. Okay, see you again.
Okay, bye for now.
All right, Assalamualaikum warahmatullahi wabarakatuh. Okay, we're back to our second session for today, the last topic for our international summer school. So hopefully everyone is getting more excited. It's going to end somehow. Anyways, okay, our speaker for today, for the last one, is actually one of the biotechnology student. So he is from the organization, the head of the organization of the faculty for the students organization, as well as the leader for this Membangun Desa project as well. So without further ado, I would like to invite Viko Adi Putra to give his experience and share to you about the virtual on the virtual tour of Ladok Nongko Salak Plantation. Okay, the time is yours, Viko.
Okay, ya, terima kasih. Hello, introduce me, Viko Adi Putra. I am student of UNISA Biotechnology. Thank you for the opportunity give here. Will share about the year of biotechnology student in building village for country. Okay, before I continue, let me share screen for Will, maybe, and friend will use some advance of new implementation biotechnology to improve the country.
Okay, dear students. Okay, so there's in this chat box, right? There's this link www.menti.com. So you can enter the link, and we're going to play a little bit of game first. All right. So everyone ready? Does any one of you enter the link? Okay, so we got one answer here: innovation and creativity, product tempeh, yogurt. What else? Okay, bioeconomy. I could see bioeconomy, reduce pollutions. Maybe there's like something more specific or anything that you might share to increase food. Okay, food security. All right. Else? Right, let's see. There's vaccine developer. Yes. Okay, more. There's bioinformatics. Okay, bioinformatics on what? Resolve food shortage. Yes, food security. And then here solve problem. Ni connectionnya kemana? Clean the environment. Okay, let's see what else here. We have produce more GMO. Okay. All right. Else? Okay. All right. Microalgae industry. Good. All right. Any more? What else here? a green world. All right. And then let's see, circular economy. Yes. All right. Research about genetics. I see. More produce medical application. Yes, medical application. All right. Okay, maybe we can move on, Viko, to the next one.
Oke, kita tunggu dulu atau lanjut, Bu?
Lanjut, nggak apa-apa.
Oke, ini ada yang masuk. Oke, I think I read to thank you, your participation. Next, kita kembali lagi ke PPT. Oke, slide 2. Oke, Agrotourism Salak Nongko is one the pioneer tourism village in Sleman Regency, Mojokerto, District Sleman, Daerah Istimewa Yogyakarta. This agrotourism provides facility for visitors and learn the cultivation of salak plant from start to finish, from the process of planting seed and care with salak grade by the export, and the processing salak of several products such as the candidate salak, geplak salak, and other processed product salak with process traditional in several local people homes. The visitor will also be preset will view the specification comfortable salak plantation area with several artificial packages facilitation. The Salak Pondok Ledanongko Agrotourism has begun experience decline is in the number of visits since the year last, especially during pandemic, the COVID-19 pandemic beginning 2020, agrotourism experienced drastic decline, especially the economic file. The government gave call for large-scale social resistance in various regions and has impact on implementation of inconsistent tourism village operational schedule. This has negative impact to community economics. Next, the Biotechnology Student Association of the University of Asia Yogyakarta received grant from the Indonesia Ministry Higher Education, Research and Technology to develop tourism village, diversifying salak processing product and government management. There is diversification of salak processing product with include yogurt salak, brown chip, and ecoenzyme along their permit. The government management effort have been started include inserting the social media for product commercial and data collection on tourism village visit and landscape designs. Next. Oke, selanjutnya ada masuk ke ini video dokumenter.
Okay, we wait for a while for the video to be played.
Sejarah awal dari desa-desa Tanokane adalah mulanya adalah perkumpulan dari para petani salak pondok di Lodoknowo ini mengadakan suatu perkumpulan, misalnya perkumpulan petani, terus itu dibuat tanaman itu dibuat edukasi untuk menarik ataupun menarik pengunjung atau tamu yang wisatawan yang datang ke sini untuk belajar maupun menikmati hasil pertanian di sini. Jadi di sini istilahnya dulunya di sini itu kan lahannya lahan pertanian. Jadi terus sejak tahun 80-an itu kan terus dimulai ditanami salak, ditanami ini. Jadi untuk pertanian kayak padi itu udah habis, diganti dengan salak yang waktu itu memang lebih menjanjikan daripada yang lain gitu. Terus kita bongkar lahan itu, kita tanami salak, sudah semuanya salak, terus sudah berbuah, terus dari para pemuka di sini mengadakan suatu perkumpulan kelompok di sini ya untuk menampung aspirasi ataupun dari para petani untuk perkembangan, misalnya untuk mengembangkan usahanya di bidang salak pondok itu supaya bisa maju, dan dengan adanya kelompok itu bisa mengakses ke dinas untuk bantuan mungkin dari pertanian atau lain. Terus sejak tahun 2005 ini kami dari kelompok pertanian si cantik Lodoknowo ini untuk dimulai kita kembangkan ke wisatanya edukasi, jadi wisata edukasi perkebunan salak di sini. Oke, untuk itu dari itu mulai saat itu saat tahun 2000 kita sudah banyak menerima tamu-tamu dari baik dari luar negeri maupun dari luar Jawa, dari Jakarta, dari mana itu kita bersini berkebun, bagaimana cara kita itu menanam pohon salak, terus itu bagaimana kita membudidayakan, kita merawat sampai hasil panen itu kita salurkan kemana itu semuanya dipelajari sebagai biro paket wisata di sini. Oke, itu selanjutnya. Jadi misalnya dulu dengan situasi wisata yang sudah maju, tiba-tiba ada COVID itu di sini wisatawan juga berhenti karena kami juga ada larangan dari dinas untuk istilahnya kontak maupun menjaga aturan-aturan dari COVID-19 itu yang harus mematuhi peraturan-peraturan, tidak boleh berkumpul, bagaimana dengan mengumpulkan orang banyak, kita harus menghindari itu dan kita harus menjaga jarak, karena itu untuk wisata pandemi itu benar-benar terpukul karena kami tidak bisa istilahnya kita menerima wisatawan dari manapun, dari lokal ataupun dari luar karena terkait dengan peraturan-peraturan itu. Maka setelah di saat pandemi itu wisatawan Danau ini hampir karena kita mentaati peraturan, makanya berhenti, jadi tidak ada kunjungan yang secara itu diadakan. Jadi untuk wisatanya memang kami tutup. Sekarang setelah ini mulai ada kelonggaran, kita buka kembali wisatawan ini untuk paket wisata kebun, untuk ini dan kita buka. Terus di sini di desa wisata ini kita juga mengembangkan di samping wisata kebun, kita juga masih mengembangkan budaya-budaya di desa, di Pedukuhan itu ada di sana ada wisata anggrek, di mana kita membudidayakan anggrek, bagaimana kita merawat ataupun kita mau tahu tentang macam-macam anggrek dan bagaimana jenisnya dan bagaimana perawatannya. Terus nanti di sana ada kolam ikan, ada sungai, itu akan kita kembangkan sebagai penunjang supaya untuk wisatawan di sini bisa gairah lagi dan mengunjungi ke sini semakin banyak lagi seperti dulu waktu sebelum Covid. Kami memang sudah banyak, misalnya ratusan ribuan orang yang wisatawan ke sini. Namun setelah pandemi ini bertahap-tahap berkurang secara drastis, namun alhamdulillah untuk saat ini sudah mulai ada pergerakan-pergerakan. Makanya kita membuat paket yang baru untuk kelanjutan supaya daya tarik dan daya saing wisata kita tidak kalah dari yang lain, dan supaya wisatawan bisa menghadiri ataupun datang ke sini untuk berwisata di wisata Danau ini.
Halo teman-teman, untuk kegiatan ini merupakan keliling di kebun salak Ladonongko. Ini mengedepankan bagaimana pengolahan salak secara organik tidak menggunakan pupuk kimia. Lalu untuk tahapan pemrosesan yang pertama ada penjarangan ataupun lalu ada saya ulangi mungkin untuk video virtual yang saya tayangkan. Ini merupakan sewaktu kunjungan dari manusia juga dari UTHM datang ke sini tepatnya di Agrowisata Salak Ladonongko dan diajak keliling, tour guide keliling perkebunan salak. Lalu untuk kegiatannya sendiri di kebun salak itu ada edukasi pertumbuhan salak mulai dari pembibitan, perawatan, sampai pemanenan dan tahap pengolahan produk dari buah salak seperti itu. Lalu di perkebunan ini di sini dari tahun 2000 ataupun 20 tahun yang lalu sudah melakukan sertifikasi organik seperti itu. Jadi ini salah satu bentuk juga implementasi dari bidang bioteknologi. Lalu selain itu untuk pengolahan produk unggulan di sini saat ini buah salak itu sebagian besar diekspor dan sebagian kecil diproduksi menjadi yogurt salak dan brunchi seperti itu. Selain itu di destinasi wisata Agro Salak Ladonongko menawarkan juga wisata edukasi berupa aktivitas yaitu pengolahan produk salak menjadi geplak salak yang diolah secara tradisional. Selain itu di desa wisata ini juga menawarkan spot kebun anggrek seperti itu yang tadi sempat terlihat ya sebentar. Lalu selain ada kebun salak, lalu ada anggrek, yang terakhir saat ini juga sedang pengembangan ikan, yaitu kolam ikan. Baik, selanjutnya Agrowisata Ladonongko ini memiliki basis yaitu kearifan lokal, berbudaya, dan aktivitas. Lalu untuk berbudayanya kita juga mengenalkan kegiatan-kegiatan yang tradisional yaitu seperti ini. Ini adalah pentas jaranan saat latihan seperti itu. Ini untuk destinasi yang terakhir yaitu pengembangan perikanan, untuk pemulihan ekonomi pasca COVID juga di sini ditawarkan juga sebagai bagian dari paket wisata Salak Ledok Nongko seperti itu. Baik, mungkin seperti itu untuk virtual tour di Agrowisata Salak Ledok Nongko.
Okay, thank you, Saudara Viko, for the presentation as well as the video. Okay, so dear students, do you have any questions regarding Salak, or maybe Viko, can you explain a little bit about how Salak plantation is, the plantation, and how to plant Salak a little bit? Maybe you can share.
Baik, terima kasih atas terima kasih atas pertanyaan yang disampaikan. Untuk kegiatan untuk pengolahan dari awal, dari pembibitan sampai tahap akhir, yaitu tahap pemanenan. Yang pertama yaitu ada pembibitan, pemilihan bibit, lalu setelah itu ada penyulaman, ataupun kadang misalkan pembibitan bersama itu tidak semuanya bisa tumbuh bareng-bareng. Jadi perlu adanya penyulaman seperti itu. Lalu selanjutnya setelah 3 bulan dilaksanakan yaitu perkawinan. Setelah perkawinan yaitu 2-3 bulan kemudian akan berbuah seperti itu. Kan ini sebenarnya untuk buah salak ini sendiri tumbuh sepanjang tahun. Lalu di Agrowisata Salak Ledok Nongko ini dari tahun 2000 atau sekitaran 22 tahun yang lalu sudah melakukan sertifikasi organik, yaitu mengedepankan perawatan tumbuhan secara organik seperti itu. Lalu selanjutnya untuk hasil buah salak saat ini itu sebagian besar dilakukan ekspor, lalu sebagian besar juga dibuat sebagai produk-produk tradisional seperti ketupat salak, jenang salak. Lalu juga ditambah lagi ada produk unggulan yaitu pencit, lalu yogurt salak seperti itu. Lalu untuk pengolahan limbah salak seperti buah salak yang sudah matang melebihi itu diolah menjadi ekoenzim seperti itu. Mungkin itu Ibu.
Oke, mungkin bisa menjelaskan sedikit tentang ekoenzim.
Oke, untuk ekoenzim sendiri, mungkin sebelumnya sudah dibahas atau belum di summer camp ini? Baik, untuk ekoenzim sendiri, itu sebenarnya dari buah-buah yang sudah masak berlebih, itu dilakukan disimpan dan dicampur dengan mol selama kurang lebih 3 bulan, lalu nantinya pengaplikasiannya sebagai penambah deterjen, pewangi ruangan, dan kegiatan pertanian seperti itu. Mungkin itu Ibu.
Okay, thank you, Viko, for the explanation. Okay, now maybe any questions from the students here? Or any one of you are interested to attend, or I mean to come to Indonesia and see this salak plantation, as well as just to come and visit us at UNISA. Okay, and also maybe the committee can share the attendance list again for the second session. Okay, yes, it's a good place to visit. All right, I think since okay.
Aaron?
Right. Okay, so I think this is the end of our international summer school for today as well, and this is the last day. So we will move on to close this program. So I would like to pass this session to our MC Marwa, so you could close the program. All right. Thank you, Marwa.
Thank you, Ms. Sarfina, for guiding us until we are here at the last day of this program. So, dear all participants, now we have come to the official closing ceremony of the Virtual International Summer School, Science and Culture, UNISA-UMP. I would like to thank you all for participating in this exciting program. You are doing great, everyone, for these 4 days. And to officially mark the closing ceremony, I would like to invite Mr. Arif Bimantara, the head of Biotechnology Study Program, to deliver his speech. To Mr. Arif, the time is yours.
Okay, thank you, Marwa. Can you hear my voice, Marwa?
Yes.
Okay, thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillahirabbil alamin. Allahumma shalli ala Muhammad wa ala alihi wa sahbihi ajma'in. Amma ba'd. The Honorable Dean of Faculty of Industrial Science and Technology, the Honorable the Head of Industrial Biotechnology Program, and the Honorable all of the speakers, all beloved workshop committees, all of workshop participants, and all of you who present today. Firstly, I would like to express my appreciation to you all, the speakers, for their excellent thought-provoking talks, all workshops committees for organizing and keeping things both under control and reasonably on time. However, most importantly, I would to thank you all, the participants. Without your input, ideas, and discussion, this workshop would not have been the success it has. Okay, this 4 days workshop is the first workshop that held by the collaboration between UMP and UNISA together. I hope that this event become a good start for the other our collaboration activity, particularly in order to strengthen the students' soft skill and hard skill, also for broaden their horizon or insight, particularly in the biotechnology application. The topic science and culture was chosen for this workshop because sometimes some researchers just focus on finding something in order to fulfill their career or other goals without caring about the impact of their research to their society or their world. So I hope this workshop will keep us remember that there are a lot of things that we can do with biotechnology to improve our life quality by implementing our knowledge, grounding this sophisticated biotechnology to our society and become one of the alternative solutions for their problems. And we apologize if during the event there are still shortcomings. I hope in the next workshop we can hold this event by offline in UMP or in UNISA, so we will have more interactive communication. I think that's all that I would like to say for this moment. Again, thank you, see you, and in the next collaboration agenda. And thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi
Waalaikumsalam warahmatullahi.
Thank you, Mr. Ari, for the remarkable speech and the official closure of our program. And with this, the virtual International Summer School Science and Culture, UNISA-UMP, is now officially closed. And now we move to the next agenda. It is review testimonials. So I want to know, what do you guys think about the whole program? Did you enjoy every section of it, or did you find it interesting or something? We are happy to hear your opinion about this event. So is there anyone wants to give some comments or?
Okay, maybe a comment from Dr. Adila as well for this program, or any words for us all.
Okay, Assalamualaikum and good afternoon to all. Okay, yeah, thank you very much for UNISA for arranging this summer school. We really appreciate it. And then, yeah, we really hope that we can further collaborate in the future. And I also hope that all the participants today will get get lots of knowledge, maybe new things for you, and also, yeah, I also learned many things here. Okay, so yeah, I hope we can continue collaborate in the future. Yeah, I think that's all for me. So thank you to all.
Thank you very much, Dr. Adila. And maybe I want to hear some of the students here who attend our closing ceremony here. May one of you from each university to represent a review or some comments on this program. I would like to invite maybe first from UMP to share. Anyone? Any one of you would like to share their comment? Please do raise hand and open your camera so that we could have a little bit of interactive with the students here. Anyone from UNISA, from UMP who wants to give a comment, maybe? Let's see, or should I call one of your names? Okay, no one yet? Okay, maybe from UNISA, please. One student. I would like to hear your comments, please. All right, Divya, may you open your camera and tell us about your experience, your comment maybe, so that we could hear your voice as well. Sorry, it's okay actually. Okay, maybe else? Okay, let me read from the comment then. So from FE Fever as well, there is a lot of creative projects that are interesting and unheard, beautifully presented throughout all, very interesting lecture from all lecturers from as well as from UMP, UNISA, so FE Fever from UMP. Any more comments from the students here from UMP? Or maybe some inputs or suggestion that we could do, or maybe for our next collaboration, you guys can actually give some comments on that. Anyone from UMP first? All right. So maybe from UNISA, please give the students give a comment or suggestions for our upcoming program as well. Okay, students. Okay, a visit to UNISA in future. Yes, you may. Maybe we can have a mobility or an exchange student program, hopefully. Else? Anyone else? Maybe from UNISA, please. All right. From UNISA, any representatives?
Baik, saya dari perwakilan dari mahasiswa UNISA sangat berterima kasih atas adanya acara summer school dari UNISA dan UMP dan berharap akan ada event-event yang lain atau bahkan mungkin UNISA bisa visit ke UMP. Terima kasih.
Yes, very welcome.
All right. Yeah, hopefully we have one time or any opportunity to go to UMP, and maybe we could do this program offline as well.
Oke, kalau dari saya, thank you for this event. Feel the holiday with the side. Jadi mengisi liburan saya dengan ilmu-ilmu terus pengetahuan yang baru. Terima kasih. Semoga depan bisa kolaborasi kembali.
All right. Yeah. All right, I think we've heard a lot of insightful comments from the students as well as from UMP, I think. Priya Darshini, it was a great virtual learning and culture experience. I have gained many insights. Thanks to the committee and members for organizing such events. All right, so I think that's all from the comments of this, the students' comments, and maybe I will also would like to inform you that the certificates for all the participants, the committee, as well as especially for the speakers will be emailed to you soon. Hopefully in, I think, 1 to 2 days time, hopefully could already get the certificates. And also I think back to Marwa to finish the session, the MC session.
Thank you, Ms. Arvina. Dear participants, ladies and gentlemen, now we have come to end of our occasion. On behalf of the organizer for Virtual International Summer School Science and Culture, UNISA-UMP, we would like to apologize for the weaknesses that we encountered throughout the event. We would also like to thank you for your understanding and cooperation. We believe that all committee members have done their very best to ensure the smooth run of their program. And once again, we would like to express our gratitude to all guests, speakers, lecturers, and all participants. Uh, before before I close this event, uh, can you guys open your camera and we can take picture together?
Okay, so everyone, please open your cameras, put your background zoom on so we could take a really nice picture, okay? I don't want to see those black backgrounds, please. It's the end of our time. We should have all participating in this, okay? All right. Okay, waiting for all of you to open the camera. I think this is better, right? To see all of your faces. It's okay if you're at home in your room, because I know it's like this is a summer school, right? So everyone should be in a holiday season, holiday mood, but all of you have to come and join us. So I really appreciate your time. Okay, more people, please open your camera so we could take a good picture together. Okay, we're waiting for some others. Yang nak pakai tudung pun boleh, yang belum pakai tudung. Yang pakai kerudung, yang belum pakai kerudung, yang cewek-cewek ini. I can speak both language fluently, okay? All right, more coming, students. I want to see more. I think we have 40 participants here, but I still only see about, let's say, not up to 20 who open their cameras. It's okay, we will wait for you all. Come on, let's go. All right, okay, I'm still waiting. The rest. Okay, should I call your name then? Okay, who haven't opened their cameras? Young Yi Liu, okay, open. Premi, Weni Arum, Dia Putri. Who is Asus? Pavitra, Suci, Tan, Ibu Ika maybe, Matumita, Santini. Okay, all right. Okay, else? Anyone else? Okay, yes. More faces. Okay, waiting just a bit more. Unless for those who cannot open their camera, it's okay. The rest? Okay, waiting for some more. I think we still have a few more. Aina, I appreciate you for opening your camera, even though you're on the way somewhere. All right, anyone else? Okay, should we start taking pictures then? Okay, committee, please take the first slide, okay? Let's count. San. All right, secondly, the second page. Siji, loro, telu. All right. Thank you all, dear students, for coming to our international program. Hope you had a great time with us, and hopefully we could see each other soon, okay? So the UMP students can see the UNISA students and vice versa. Both of us can go to both places. So yeah, that's it. Have a great holiday and hope to see you back to your respective campus soon in October. Okay, that is all for our International Summer School Program. See you again another time. All right, bye. Assalamualaikum warahmatullahi wabarakatuh.